for Chapter 3, Section 4, Diffusion and Osmosis. So far, we've looked at the cell theory. We have walked through each of these various organelles that we find in eukaryotic cells. And in the last recording, we looked at the cell membrane and its very specialized structures that allow it to perform its tasks. Now today, we're going to look at diffusion and osmosis, one of the primary processes that has to occur for every cell. Let's make this larger. All right, diffusion is a passive transport mechanism. That means that things are able to move across the cell membrane without the cell expending any energy. It doesn't cost the cell anything for a molecule to diffuse from one side of the membrane to the other. This passive transport mechanism is always going to go in the direction from high concentration to low concentration. Every time. Since diffusion doesn't use energy, that's the only direction molecules can move. So in this example here, we can see the beaker of water has one drop of green food coloring in it. Well, the food coloring is very highly concentrated right here in this spot. If we go farther to the edge of the beaker, there's a very low or no concentration of the green food coloring. In this region, we can see there's a bit more. So this is like a medium concentration. This is a very high concentration. So this green food coloring is going to spread out in the water, meaning it's going to diffuse through the water from where it's concentrated, and it's going to fill all the spaces where it's not concentrated. Osmosis is another kind of passive transport, but it's a little specific. It only describes the movement of water across a semipermeable membrane. Now if we take a look at three cells here, the movement of water that we see in osmosis is going to be dependent upon the solution that this cell finds itself in. So if I take this red blood cell, which they always have this shape, and I put it down into a beaker of some solution, and the water is able to move from the outside to the inside, back and forth at a steady rate, at the same rate it's going out, the same rate it's coming in, then I can say that this cell is in an isotonic solution. So the movement of water is balanced in and out of the cell. If the cell is placed in a solution and it constricts or it shrinks, what's happening is the water inside the cell is being drawn out. It's moving out of the cell more than it's moving in. This kind of a solution is called a hypertonic solution. A hypertonic solution, like hyper kids get kicked out of class, well, a hypertonic solution causes water to come out of the cell, causing the cell to shrivel and sometimes even die. If a cell is placed in a solution and then all the water rushes in and the cell swells up to great size, almost to the point of bursting, this cell is in what's called a hypotonic solution. The solution outside the cell has more water than the inside of the cell does, so the water starts to rush in. The water always moves from high concentration to low. So in the hypotonic solution, the water is more highly concentrated out here. So it flows from high to low concentration. In the hypertonic solution, out here there's less water. There's more water inside the cell, and so the water moves from high concentration to low concentration, causing the cell to shrivel. The last type of movement we're going to look at today is called facilitated diffusion. Now, water and small molecules are able to just pass back and forth across the cell membrane, no trouble. They just go back and forth between the phospholipids on their own. But there are some molecules that need to move across the membrane. Either it's something the cell needs to have on the inside, or it's some molecule that needs to get out that can't pass through this middle region. Maybe the molecule is too large, or maybe it has a charge, a positive or a negative charge. Charged molecules can't go through these fatty acid tails region. It gets pushed out like a magnet. So if we have a large or a polar molecule, it's going to have to use another mechanism. And in this case, we're going to look at a transport protein. This blue structure here is just a protein that's embedded in the cell membrane. It's stuck in here. and it's basically acting like a doorway. It allows a specific molecule to pass through the protein, so it doesn't have to touch these fatty acid tails at all. It's protected from the nonpolar region, so this molecule can just pass straight on through to the inside. Now, if a molecule is really large, it needs this larger opening to get through. These proteins are very, very specific on what molecules they will allow to pass. This one only works with the little blue circles. If there were some red squares or some yellow triangle molecules here, they couldn't go through. This protein is only for this kind of molecule. So since there are hundreds of different kinds of molecules that have to be able to pass back and forth across the membrane, in every cell membrane there are hundreds of different kinds of membrane proteins allowing one kind of molecule to pass in and to pass out through that particular protein. They're very specific, so they have to function properly in order for that cell to function properly. The next section is going to be on active transport. We will get to that in our last recording.